Hi, I'm going to teach you about DNA replication. I'm not going to go into very much detail because I just want to give you a visual demonstration. Here's my DNA molecule, and I hope you appreciate how ridiculously long it took me to make this thing. Anyway, DNA replication starts when enzymes start to break apart the DNA molecule at what we call a promoter sequence. Remember that the bases are held together by hydrogen bonds, which are very easy to break apart. This breaks apart, and at the point where it is being broken apart, we call that the replication fork. An enzyme called DNA polymerase adds new nucleotides to the unpaired nitrogen bases that are exposed at the replication fork. It can only do this in the 5' to 3' direction. So if this is the strand that runs in 5' to 3', it just starts at the end and adds new nucleotides. Adenine pairs with thymine, guanine with cytosine, thymine with adenine, cytosine with guanine, thymine with adenine, so on and so forth. Remember that the two strands run anti-parallel to each other. What that means is that if this strand is running from the 5' to 3' direction, then this strand is going to be running from the 3' to 5' direction. However, new nucleic acids can only be added in the 5' to 3' direction. So how are we going to add new nucleic acids in the 3' to 5' direction when we have to start from here and go this way? What happens is that the new nucleotides are added in segments. We call these segments Okazaki fragments. And this is how it works. Move this out of the way. DNA polymerase comes in and it has to add from this direction to this side. And so as it opens up, DNA polymerase is going to come in and add an adenine here, a cytosine here, and a thymine here. As the replication fork moves down the molecule and more bases become exposed, DNA polymerase will continue adding new nucleotides in the 5' to 3' direction. So now it will add an adenine here, an adenine here, and a guanine here. We call these fragments that it adds new nucleotides in Okazaki fragments. New nucleotides are added simultaneously to both the leading strand, or the 5' to 3' strand, and the lagging strand, or the 3' to 5' strand. This continues down the DNA molecule until you have two separate DNA molecules. Notice that each new DNA molecule has one old strand and one new strand. This is called semi-conservative replication.